So yeah, welcome. This is an IHC workshop for the post-mortem brain tissue. And I'll start with a quick company introduction before introducing the speakers who will then take over and lead the webinar. So in case you aren't aware of who we are as a company, Proteintech is a leading life science reagent company, and we have products which span across 3 core brands. Through our Proteintech brand, we sell our antibodies and immunoassays, and this is definitely what we're most well known for. We have antibodies against 13,000 human targets, as well as ELISA kits, and an increasingly popular range of secondary antibodies. Through our humankind brand, we sell our recombinant cytokines and growth factors, which are used in things like iPSC differentiation and also cell and gene therapies, because we have them in GMP grade, as well as RUO. And then through our Chromatech brand, these are our nanobody-based reagents. As the name suggests, they are a much smaller antibody, and so they're excellent for things like super resolution microscopy, as well as immunoprecipitation when tagged to a bead. As a company, we say we are founded by scientists, and we're for scientists as well. So what we mean by that is that we have people like myself, PhD level, technical support, product managers, all there aiming to serve you, the customer. We have the largest in-house portfolio of antibodies for the human proteome, as well as a really stringent level of product validation for all our antibodies and other products. We also have won many awards over the time, most recently being a runner-up for the Researcher's Choice Award. This wasn't anything that was bought or paid for. This was something that was volunteered. We were voted for by our customers because of our products, but also because of things like these webinars that we put on for everyone and helping educate people in not just immunoassays, but also careers and other workshops like that. As a company, we celebrate our scientific success by celebrating our customers' success, and we're thrilled to have been published in over 250,000 times worldwide, which is now making us the fastest growing cited and product citation company in the world. And also we've now moved into the top 10 cited global antibody companies. So we're really excited about that and to see where we go in the future. So without further ado, I will now introduce our speakers. To start with, we're going to have the 2 senior research technicians from the Manchester Brain Bank. And Manchester Brain Bank will also be introduced during their talk, but essentially what it is it's a subset of the University of Manchester based in the UK, and they have they collect patient tissue for processing in neuropathology, and they also have a bank of this tissue as well for research that can be used down the line, and that will be better explained by the speakers today. So first up, we have James and Yvonne. So James is a senior research technician. He joined the Manchester Brain Bank in 2019, and prior to that, he worked at the Salford Royal Hospital, with much experience in cellular pathology. He has a lot of experience in microtomy and IHC, and his role is to process the brain tissue prior to staining. Alongside James, we'll have Yvonne speaking, and Yvonne has been working at the Brain Bank since its inception in 1986. She is also a senior research technician, and she performs the histology and IHC techniques in order to stain the brain tissue prior to neuropathological diagnoses. And both James and Yvonne have been on many papers since they joined the Manchester Brain Bank. Following James and Yvonne, we'll then have a short talk by Dr. Andrew Robinson. Andy is the manager of the Manchester Brain Bank, and he has worked there since 2009, close to Professor David Mann, who was the founder. And since then, he's been able to build his career and also completed his PhD in 2016. As the manager of the Brain Bank, he carries out large-scale dissections of brain donations, and he also assists with the diagnostics, using his extensive knowledge that he's gained over the years with his experiences of neurodegenerative disease and neuropathology. He's also an active researcher and now has over 75 publications in the neurodegeneration disease field. And with that, I will now hand over to James and Yvonne, and they will present their talk before we have a small talk from Dr. Robinson. Thank you very much.
Thanks, Rebecca. Just share my screen. Here we go. Is that okay? Yep.
Yeah, looks good.
Good. Amazing. Morning, everyone, or afternoon, wherever you are. I'm James Minshull. I'm research tech at Manchester Brain Bank, and this is Yvonne, despite my name tag just having me on. We're both here. So I'm just going to give a short agenda for what we're going to talk about today. So Yvonne and I are going to give some of the technical considerations around tissue processing, immunohistochemistry and immunofluorescence, with particular focus on post-mortem tissue. Then Andy is going to talk to you about one of our past research projects to use some of these techniques, and then we'll go to a Q&A. So what exactly is the histology process? Some of you might be more familiar with that than others, but basically it's a technique for looking at tissue. If you've had maybe a colonoscopy or more other routine operations, maybe you've had an appendix or gallbladder removed, any tissue that's ever taken into your body, that always goes to either the NHS in the UK or the health system, wherever you are, and that will be screened using histology. It's one of the best ways to look at tissue and see the morphology, look at the cells that are there, and is very important for doing cancer diagnoses. So that starts with dissection, really, from a person's body, but then as we do in the brain bank, we get whole fresh brains. They're fixed in formalin for at least 2 weeks. They are dissected by either Andy here or our pathologist, Professor Federico Roncaroli, and then they are processed to wax. They're infused with wax. They're embedded in molds to give them a bit of structure and support. And then they undergo microtomy, where thin sections are taken from them. And then they undergo staining. This pink purple stain you see here is the most common one you will see in histology, which stands for hematoxylin and eosin, which is H&E, you'll see it referred to as. And then down here, you have the brown stain, which is IHC, immunohistochemistry, which Yvonne will talk to you a bit more about later. So the first thing to do is process our tissue. So it's been dissected by a pathologist according to a pro forma that's used by all brain banks in the UK. And they go into a cassette like this, which has a case identifier and then a block number. And every block with an A1 on it, for every brain in our brain bank, will be frontal. Every block says A10 will be hippocampus. So it's very regimented. They're processed by the processor. You see these bottles down here, they contain these reagents in this table on the right. So they start in formalin, which is water-based, and basically we want to get them to wax. And water and wax do not mix. So we need to remove the water first. So we take them through these rising concentrations of IMS, that's industrial methylated spirits, or alcohol, basically. That's about 8 hours in IMS. And then IMS doesn't mix with wax either. So we need to get rid of the IMS. So we go into xylene, which is an organic solvent. It's in there for about 5.5 hours. And then finally, the tissue goes into molten paraffin wax. It's heated to about 55 degrees Celsius. And it's put under vacuum, and that pulls any of the old chemicals out, and it infuses that tissue with wax, which creates more stable tissue that can last basically indefinitely. But it can be a little bit brittle at this stage, so we need to provide a bit of support, which is where embedding comes in. So I've got a little video here. I'm going to sort of narrate it for you. So you can see here the cassette with a piece of processed tissue inside. You have a mold filled with wax. Put the tissue in, push down on this cold plate, press down to get flat against the mold. The cassette goes back in, bit more wax on top, and then that goes over to a cold plate to set. I'll play that one more time in case you missed it. So the cassette here, you see, gets pushed back onto the top of the block. So that identifier that we looked at before, that is sealed together with the tissue. And then that creates, explain again, that creates a block here, you see, is one single piece with the tissue embedded inside. I have a bone to pick with this video, in that she uses, or he uses forceps to push down this tissue here. I would advise against that, to be honest. I would use a tamping tool, which is what you can see up here at the top. It's a lot flatter, and brain especially, and post-mortem brain can be quite fragile. So it's important to be firm, but don't be too hard. You have to still be gentle with this tissue even at this stage. And why do we do this? We are surrounding that tissue with extra wax to provide support during macrotomy. Before we move to the next slide, just pay attention to this block here. You can see the tissue is inside, but you can see there's wax covering it still. So in the first step of microtomy is to remove that first layer of wax. We call this trimming. And we cut out at around 10 microns, taking 10 microns off at a time until we are what we call full face. So that's the whole of the tissue in the block is at the surface. We cool, partially rehydrate the blocks in crushed ice, and then we section, take off thin sections of tissue at between 3 and 6 microns. These sections come off the microtome, they're usually quite wrinkly, so they go into a water bath at just below the melting point of the wax, so about 50 degrees Celsius. And those sections then, at that temperature, will spread out, release all those wrinkles. You can then pick up the sections on a coated slide and they're dried overnight. And then that process, I'll show you here. So they're taking the sections off, they're turning the wheel over there on the far end. Just rewind that and jumped a bit. Sections coming off, move into the water bath. All the wrinkles have stretched out, and then she will pick up with a slide. And then that will go in the oven overnight, and that removes all of the water. It's very important. And it makes sure that the sections are completely stuck to the glass, annealed is the official term. And that makes sure that during staining, which is the next process, that they don't float off, basically. And this is what you will get as an end result. So some tips and tricks for you. So the most important thing is to keep your wax blocks cold. And crushed ice is the best. You want a bit of water in there to partially rehydrate. But you need to make sure they don't get too cold because they can sort of shatter as you cut in. So you can warm them in the palm of your hand or quickly dip them in your water bath. And also cool your sections. The blade is metal, it holds the heat, and that will impart some of the heat as you are cutting. So if you blow on the sections as they come off the microtome, that will help you a lot as well. And a problem that you probably come across a lot with post-mortem brains is what we call exploding sections. So these high lipid tissues, such as brain, this is also very common with breast, tend to do this. They will, as the melting point of those lipids is lower than that of the wax, they will just spread out and spread and spread until they just disintegrate on your water bath. And the easiest thing to do there is cool down your water bath or cut slightly thicker. I've heard there's also, you can put a bit of dish soap in your water, but I wouldn't do that. I've not tested that myself, and it may interfere with your staining. And once you have your lovely sections on your slides, you can then move on to immunohistochemistry, which Yvonne will give you a rundown on next.
Excuse us while we just swap over. Hi everyone. Thanks James for that. I'll be handing you back to James in a little while so that he can talk you through immunofluorescence. But first, I'd like to take you through the process of immunohistochemistry that we frequently carry out here at Manchester Brain Bank. And I'm just going to go through the basic procedure, highlighting the steps that we use for post-mortem formalin fixed brain tissue, the equipment, the potential pitfalls, hopefully some of the solutions as well. And I'm hoping that at the end of my part of this webinar, that even if you're a complete newbie to immunohistochemistry, you'll have the knowledge and the confidence that with the help of a kitchen appliance and a box of chocolates, you can successfully carry out immunohistochemistry for yourselves. So I have probably the best job in Manchester Brain Bank. I take this dull, unstained section that James has given me, and I turn it into something that can be diagnosed, captured, qualified, quantified, and studied for years to come. So first of all, what are the basics of immunohistochemistry? If you've never done it before, then I'm sure that Rebecca has links to some excellent resources and webinars that she can give you at the end of this talk. But first of all, the theory. So we start off with a slide looking at our antigen of interest, or epitope. So the epitope is just the part of the antibody, the part of the antigen that the antibody sticks to. So we start off with a primary antibody. An example of what we might use here in the brain bank is a mouse monoclonal antibody to beta amyloid, say. We then add a biotinylated secondary antibody. So the secondary antibody might be raised in something like horse against mouse. And we would hope that the only place that that would stick to on our slide would be where the primary antibody is. There shouldn't be any other mouse anywhere else in that brain. The secondary antibody is biotinylated, and biotin is sticky. It's sticky to avidin. And so the third step is we put in an avidin biotin complex, and this multiplies everything up and makes it a big blob. But the avidin biotin complex is also linked to an enzyme. And in our case, the enzyme we commonly use is peroxidase. Peroxidase has a substrate, DAB, which you can see in number 4, and that is diaminobenzidine. And that forms a brown precipitate in the presence of peroxidase. So that's the theory. But how does it look like in practice? Well, we start with James's waxy section on a slide. And the first thing that we have to do is pretty much undo absolutely everything that's happened to the tissue so far. So the start of this process is to remove the wax and return the section to its aqueous state. And we used to do this manually in a fume cupboard with lots of different jars. But thanks to an unwanted piece of equipment, we now actually have an auto stainer that does this automatically for us. So what's the auto stainer doing? Briefly, the tissue is first of all warmed to 65 degrees. 5 minutes. That didn't used to happen when we did it in the fume cupboard, but the auto stainer does it, which is good. The slides are then incubated in xylene. As James said, xylene is solvent for wax. That gets rid of the wax. Another wash again that gets rid of any waxy residual xylene. This wax removal step is really important. And if you're getting low levels of staining for some antibodies, then probably the best thing and simplest thing you can try is to increase this time of dewaxing. And in fact, we've increased our time over the years. So as you can see, we currently use 2 lots of 10 minutes to dewax. The slides then go into industrial methylated spirit. 2 washes. We used to use absolute alcohol, ethanol, but it's too expensive, so we use IMS now, does the same job. And then we gradually introduce water to the section. Don't want to osmotically shock it, so 10% water, 30% water, and then finally we go into tap water. So the next step is antigen retrieval, or also called antigen unmasking. And this is especially important for formalin-fixed tissue. As James said, the tissue is in formalin for a long time before it gets to this point. And that formalin has caused a lot of cross-linking, which actually forms a physical barrier to stop the antibody effectively binding to the antigen of interest. So there's several methods of antigen retrieval. And if you look at the data sheet that comes with your antibody of choice, we'll have some information about that. So, for example, our Proteintech TDP43 antibody recommends TE buffer, tris-EDTA buffer at pH 9, or they also say you can use citrate buffer at pH 6. Well, we routinely use citrate buffer, so that's what we use. You can buy it, but it's easy to make. And this method is called heat-induced epitope retrieval, or HIER. And it involves incubating the sections in citrate buffer or TE buffer at around boiling point for about 20 minutes. For our heat source, we originally used to use a microwave for many years, but over the time there were reports that microwaves cause hotspots in the tissue and lead to uneven staining. Now, in actual fact, that never seems to be a problem for us, but we did eventually stop using it, and we got ourselves a pressure cooker, We bought a proper digitally controlled pressure cooker that was called an antigen unmasking unit, and it worked very well. But it was very expensive and it broke down after every service. So 2 pressure cookers and many thousands of pounds later, we gave it up and we went back to the microwave. But then our current amazing professor pathologist came along and he said, this is what you need. And he produced a supermarket vegetable steamer that cost £10. It works brilliantly and I would highly recommend it. It's robust, it's reliable, it's cheap, and we get very good results. We use these little containers inside it, but any heat safe, liquid safe, lidded slide holders will do the same job. After antigen unmasking by this method for our citrate buffer, we cool the containers plus the slides and the buffer under cold running tap water for 10 minutes. And apparently this gradual cooling process is also an important part of antigen unmasking by HIER. And then, as ever, we wash our sections in water. But apart from hot citrate or TE buffer, there are a couple of other antigen unmasking methods. There's enzymatic reaction, so you can use proteinase K or trypsin. People do, but we don't. But 2 of the antibodies that we routinely use here at Manchester Brain Bank recommend formic acid pre-treatment as a means of antigen unmasking. And for those antibodies, we incubate in 70% formic acid for 20 minutes. And again, afterwards, we wash after the incubation. The next stage that we go on is to quench any endogenous peroxidase activity on the slide so that we don't get any false positive results. As you can see in the diagram, our substrate DAB precipitates in the presence of peroxidase, but there is peroxidase activity, especially in brain tissue, especially around the blood vessels. But we only want our DAB to precipitate where the peroxidase is associated with the antigen. So it's important that we quench or use up any endogenous peroxidase by saturating it with hydrogen peroxide. So we incubate our sections with 3% hydrogen peroxide in methanol for 30 minutes, followed by washing in water. And then at this stage, we take our slides out of the slide holders and place them on slide trays. It's very important that the sections don't dry out, as this might increase non-specific binding. Now, our slide trays are very low tech. We found the best ones are plastic chocolate boxes with lids, cut down thin pipettes and layers of damp tissue supporting the slides. And then all subsequent steps up to the DAB incubation are carried out in these little humid chambers. The final step of the preparation before we add the primary antibody is first to add a hydrophobic barrier around the section. And this ensures that any reagents we subsequently use will actually stay on the section and not wander all over the slide. It's important when antibodies and kits are so expensive or they're in short supply or both. A little tip I'd say is to make sure that the buffer doesn't touch the hydrophobic barrier until the pat pen or image pen or whatever it is you've used has dried a little bit, because otherwise you might get an oily layer that floats on top of the buffer, and then when you pour that off, it sticks onto the section and creates patchy staining. Once it's dried a little bit, the image pen or whatever you use, the liquid can go right up to that. So this is followed by the blocking buffer step, which involves incubating the sections for 30 minutes at room temperature or overnight at 4 degrees in a protein blocking reagent. The theory is that if you use serum, it contains antibodies that bind to and block any non-specific sites. So it minimises any non-specific binding between your antibody of choice and your sections. We use secondary antibody species serum. So in the example before, we use horse serum to block, because that's what's included in our commercial kits. So finally, we can actually get to the primary antibody. And from then on, the process is pretty much as it is in the diagram. For the primary antibody incubation, hopefully, you'll get a data sheet with the antibody, which will give you a ballpark figure of where to start from in terms of dilution. But if you've absolutely no idea about where to start or no information, then I would say 1 in 200 is as good a place as any to start. But it's always worth trying a few dilutions either side of that or the dilution that you've been recommended before you start. So we wash, on with the biotinylated secondary, wash, on with the avidin biotin complex, wash, on with the DAB. All our washing steps are exactly the same, phosphate buffered saline, 3 changes over 5 minutes between each step. Step 4, the chromogen, DAB in our case. The chromogen is just a name for a coloured product that you can see under a microscope. You might read about monitoring DAB colour formation under the microscope until you get the desired staining intensity. We don't do that. We standardise this step 4, at 5 minutes per section. There's a couple of reasons for that. One is that DAB is nasty and we don't really want it floating all over the lab and all over the microscope. And more importantly is that if we've got 2 sections side by side that we want to compare, we want to know the intensity of staining reflects the antigen load, not the fact that I left 1 in DAB for 2 minutes and 1 in DAB for 20 minutes. The method is lovely. It's foolproof. You just have to make sure you're using the right kit, so the right secondary antibody for your primary antibody. You're using the right enzyme for the right enzyme substrate for the enzyme that you're using. But really, if you've got all that under control, you can't go wrong. Following the chromogen formation, the colour, we counterstain our sections with hematoxylin, and that stains nuclear material pale blue. And this is important because it helps us characterise the staining. Is it nuclear? Is it cellular? Is it extracellular? And it also helps to identify where the heck you are in the brain if there is no IHC staining as well. So in order to preserve the slide, we go through the whole dehydration process again, the exact reverse of what we did before, and more or less what James did the first time round. Again, we use the auto stainer, but instead of going from xylene to wax, as James did, we're going from xylene to DPX. And that's because DPX has the same optical density as glass. So when you're looking at it and focusing through a section, you don't get any aberrations. It forms a permanent seal between the section and the cover slip, and it preserves the sections forever, for a long time. Again, we used to do the cover slip manually, but now we have a cover slipper that actually does it quicker and a lot better than I can. So what problems do we get with immunohistochemistry? Most of the problems are to do with either high background staining or low antigen staining. As mentioned before, if you've got low antigen staining, the first thing to try, the easiest thing, is to increase the time of dewaxing in xylene. Failing that, you can also change your antigen unmasking method, but always check your primary antibody concentration, always optimize that every time you change your conditions. If the background staining is high, you can try different wash buffers. You can add a detergent to your wash buffers such as Tween, but again, always check that antibody concentration. It's always good practice to include a positive and a negative control wherever possible. The positive control should be a tissue that you know contains the antigen you're looking for. There's many different negative controls that you can do, but the basic negative control should be treated in exactly the same way as all the other stained sections, but with the omission of primary antibody. And that's exactly what I do. I incubate the negative control just in the blocking buffer while everything else is in primary antibody. In my experience, if you've tried all this and changed all these things and you're still getting rubbish results, the chances are it's going to be your antibody that you're using. So if at all possible, try an alternative antibody. In terms of shortcuts with the whole of this procedure, in all honesty, I don't take any. I don't cut down the times of incubations. I don't cut down the numbers of washes. The method is very robust in that the incubation times are not to the minute, with the exception of DAB, but nevertheless, I try to be as consistent as possible. So to summarise, Immunohistochemistry on formalin-fixed paraffin-embedded post-mortem brain tissue is easy. It's robust, and it can be very low-tech indeed, and you do need to eat the chocolates to release the box. I'm going to hand back over to James, who will talk to you about immunofluorescence.
So I'm back again. Thank you, Yvonne. So immunofluorescence, it's very similar in a lot of ways to IHC. So you've got both primary and secondary. So primary is when your primary antibody that is recognizing your epitope of interest, that is directly conjugated to a fluorophore. And then secondary is very similar to what Yvonne has just described to you. So we have your primary and then a secondary antibody that itself is conjugated with the fluorophore. And each of these fluorophores is manufactured in a way that they have very specific spectra, whereby they have an absorption spectrum. So, for example, Elexfluor 568, which gives this orangey colour, or red, depending on how you set up your microscope. That is the spectrum under which it absorbs energy, and then it will release at the wavelength of this more solid peak on the right. So you can excite it at one wavelength, and then you receive a signal at a different one. And then you can, with filters in a microscope, you can look at that with your own eyes, or you can use a fluorescent slide scanner using more complex lasers, things like that. Okay, so what are the pros and cons of immunofluorescence? So one of the big pros is the fact that you can multiplex. And so that means you can use different species and different actual fluorophore combinations to produce different colors. So you can stain, if you see this picture at the bottom, you can stain one protein in green, another one in red. And if you merge them, you can see where there is a yellow signal. That is where these two proteins are co-expressed. And that can be done actually with IHC, but it's a bit more difficult to deconvolve the colors. So if you're doing co-expression experiments, it's usually better to use fluorescence, but that is changing as technology progresses. Fluorescence has higher sensitivity. It can detect proteins that are in very low abundance. What are the cons? The fluorophores can actually lose their ability to fluoresce over time. They fade, and they can also be bleached. So if you stay looking at one section of your section for a long period of time, and you're using a lot of light to get back that signal, you actually start to bleach the fluorophores. So these stains don't last forever, basically. And also particularly relevant to us in the post-mortem world, some cellular components can fluoresce naturally, autofluorescence as it's called. And there's a protein called lipofuscin, which especially generates in older people. So that's a particular problem for us if we're using that post-mortem brain tissue. Red blood cells as well can be quite problematic. So I have been optimizing some immunofluorescence recently, and I've actually been using one of Proteintech's products, this nanobody secondary. So these are generated from camelids, which are a species that include camels, obviously, in the name, alpacas, llamas, and they are very unique antibodies that the variable domain, that bit which recognizes the epitope on the antigen, is very small. And what Proteintech have done is they've recombinantly reproduced that little bit of that variable domain. And that as a whole can recognize your primary antibody. It's been conjugated with these fluorophores. And there are many advantages to this. The reduced size means they have greater tissue penetration. They have high affinity and low background. And as you can see here, each one of these, 4 of them attach to this primary, and each one has 2 fluorophores each, so it's much greater signal. And also, if you compare to this, which is the traditional route where you have a full IgG antibody, that's usually from a rabbit or a mouse, the fluorophore itself is actually quite a fair distance from the epitope itself, whereas this would be a lot closer, so it has greater resolution as well. So the actual IF process is quite similar to IHC. We dewax and rehydrate in an identical way to IHC that Yvonne demonstrated before. Antigen retrieval, I usually use exactly the same as Yvonne does with pH 6 citrate buffer, 20 minutes. And the quenching peroxidase stage, we've no need for that. We're not using peroxidase enzymes in immunofluorescence. We can get rid of that. In its place, I do a permeabilization step, if that's how to say, where you mix a bit of detergent, so Tween or Triton, with a bit of buffer, and that helps your the following steps to really get deep into your tissue. Blocking, I use bovine serum albumin or BSA. That's recommended by by Proteintech themselves. But I suspect it may be because alpaca serum is quite hard to get your hands on. Your primary is as normal, that is overnight at 4 degrees, so slightly different there. Secondary is at room temperature for an hour, and these are both mixed with blocking buffer. ABC amplification, not relevant. DAB, not relevant. But we do need a counter stain. So Yvonne used hematoxylin in IHC. That's not going to work here. We need something that fluoresces. So we use DAPI, which binds to DNA, and that fluoresces blue. And then we cover slip. There's no need to dehydrate again, go back to xylene with this. We just wash them in buffer again, and we use specially made mountants that sometimes can have anti-fade chemicals in and chemicals that prevent bleaching. So I'm just going to show you one of the recent experiments that I've done. So this is neurofilament heavy chain, and this tissue here is from the optic chiasm. So that's where the two optic nerves cross. So this is a lot of white matter. So neurofilament stains for the neural cytoskeleton. So you should be able to see, if I zoom in here, we can see these axons crisscrossing here. So with fluorescence, I always optimize with IHC first. Make sure that primary is working before you waste all your time doing fluorescence. And you make sure it follows the expected staining pattern according to the literature, according to your data sheet. So that's in terms of the cellular compartment, things like that. And make sure you've got a low background. So if you look in this vessel here, you can see it's quite clean. And then moving forwards, I'd then use the optimized primary concentration. So that here was 1 in 1,000 to then start on your immunofluorescence. And in that case, I followed my IF procedure and I got these images. So this is a nanobody against a mouse antibody with the Alexaflor 568 conjugated to it. And you can see the staining pattern is very, very similar. And this can undergo further optimization. As you can see here, in these vessels, the red blood cells are fluorescing quite strongly. So that is autofluorescence at work there. So there's further optimization. You can use things like Sudan black to reduce autofluorescence. Before I move on, I remembered I forgot to tell you something. So this IF process, once you get to this secondary antibody here, this is your nanosecondary. If you're using a conventional fluorescent secondary, make sure from this step onwards, you're always doing things in the dark. So you can use a dark room or you can cover your humidity chambers in tin foil. Yes, again. And also important to say is that fluorescence isn't just for these two-dimensional slices of tissue. You can actually take large chunks of tissue, make them transparent through something called clearing, stain with your fluorescent antibody, and then using a confocal microscope or a light sheet microscope, you can image layer by layer and reconstruct these representations in 3D of your fluorescent proteins in situ. And so in summary of this technical part of the session today. Processing longer is better for brain samples. It's very important. It's about a 20-hour process that we use. Embedding, you have to be gentle, firm enough to get it flat, but please still be gentle. You don't want to destroy your tissue. And then microtomy, keep things cool. Immunohistochemistry, Yvonne's showed you there's no shortcuts. Make sure you're doing all your rinses, making sure that you do full 30-minute rather than 30-hour incubations, and don't let your slides dry out. And fluorescence, always check your primary works first, don't waste your time. And it's always best performed in the dark from your secondary onwards. Thank you very much. That is our section for today, and I will hand over to Andy, who can show you how we've put some of these techniques to work in our research here at Manchester Brain Bank. Thank you.
Brilliant. Thanks, James. Could you just stop sharing your screen?
Yeah, no problem.
So I just share mine. Can everyone see that okay?
It's good.
Excellent. So I think you'll agree that was an amazing summary of how to do immunohistochemistry. We're very lucky here at Manchester Brain Bank to have two absolutely superb senior research technicians that have the correct protocols and the correct ideas so that we get the best results at the end of it. So my name is Andy Robinson. I'm the manager at Manchester Brain Bank. As Rebecca said at the beginning, we're based at a hospital site in Salford at Salford Royal Hospital, but we're actually a part of the University of Manchester. And based on what you've heard from James and Yvonne, I thought it'd be good to give you a flavour of how these immunohistochemical techniques work in practice. And as this is a technical seminar, I thought I'd share a recent technical paper we published called Retrospective Neuropathological Diagnosis of TDP-43 Proteinopathies, the factors affecting immunoreactivity of phosphorylated TDP-43 in fixed post-mortem brain tissue. A lovely, short, snappy title. I'm sure you'll agree. But before I begin, I thought I'd give you a little bit of a background about Manchester Brain Bank. So we were established in the early 1980s by Professor David Mann. And David's research interests were based in neurodegenerative diseases. So he set up the bank as a repository for brains from people who suffered with these particular types of disease. We currently hold about 1,000, just over 1,000 brain donations. Alzheimer's disease is the most commonly represented disease that we hold, which kind of makes sense because it's the most common form of dementia. And Manchester Brain Bank is well known for its work on frontotemporal dementia, so we hold a large number of FTLD cases. Alongside these cases, we hold tissue from rarer diseases like progressive supranuclear palsy, cortical basal degeneration, multiple system atrophy, etc., etc. We also hold tissue from a number of aged control subjects. And something that isn't on the slide is we've recently branched out into some neuro-oncology as well. So we hold 25, 30 brains now of people that passed away with some sort of glioma. We have research ethics committee approval to release tissue to external research projects without the need for UK researchers to apply for any extra approval themselves. Anyone that's overseas, unfortunately, you need to have your own ethical approval, and we would need to see that before we released tissue for your work. And here is my email address and the Brain Bank website, so you can look us up if you're interested in accessing any human brain tissue for your research. Obviously, you might not be able to scribble it down now, but you can always watch it back on the video, or you can type Manchester Brain Bank into Google, and we're the first hit there. So now back to the matter at hand. We published this technical paper back in August 2023, in the journal Neuropathology. To give you a bit of background on phosphorylated TDP-43, the pathological inclusions are found in a number of different diseases, but are mainly associated with amyotrophic lateral sclerosis, ALS, and frontotemporal lobar degeneration. And abnormal phospho-TDP-43 can be found in other diseases as well, such as Alzheimer's disease, and can also occur as a part of quote-unquote normal aging. The most common phosphorylation sites for TDP-43 are at serine 409 and serine 410. There's also been a possible pathogenic role that's been suggested for phosphorylation sites at serine 396, serine 379, and serine 403, 404. And when new pathological criteria are released for TDP-43 proteinopathies, brain banks have to revisit old cases which have been stored in formalin or as formalin fixed paraffin embedded, or FFPE for short, blocks and slides for decades, and we might need to update their diagnoses. For example, in 2019, Peter Nelson and colleagues published a new set of criteria for the neuropathological changes associated with limbic predominant age-related TDP-43 encephalopathy, or LATE for short. So brain banks had to go back to old tissue to update the diagnoses of cases that have some sorts of TDP-43 proteinopathy to make sure that the diagnoses are as accurate as possible. And as I mentioned, brain banks generally store tissue in formalin or as FFPE blocks. After initial sampling for the diagnostic side of things, brains are then kept in formalin for decades and are resampled for research when the original diagnostic blocks are exhausted. Now, we know prolonged formalin fixation can damage some antigens, leading to a reduction in the intensity of staining, whereas other antigens like phosphorylated tau, for example, that's less affected. But the effect of long-term storage as FFPE blocks and prolonged fixation in formalin on the dissection of phospho-TDP has not been studied. So in this particular work, we attempted to glean an insight into these effects and ask the question, does prolonged fixation and/or long-term storage as FFPE impact immunohistochemical identification of phospho-TDP-43? So the methods we used to try and answer this question were pretty simple, really. We examined archived FFPE tissue from 5 cases with TDP-43 pathology, and we later found out that these were late cases. And these were processed and embedded when they first came into the brain bank 11 to 13 years earlier. We also looked at tissue from 5 cases of FTLD-TDP that were donated to the Manchester Brain Bank between 2001 and 2005. And here we examined tissue from FFPE blocks processed and embedded after a short fixation, so the diagnostic blocks, so to speak, and also tissue that had been kept in formalin for many years. From each late case, sections were cut from the original diagnostic blocks of the hippocampus, the amygdala, the middle frontal gyrus, and these were chosen as they're proposed in the consensus pathological classification scheme for late. And from each FTLD-TDP case, the hippocampus and the superior frontal gyrus were resampled after 19 to 21 years in formalin, and these were processed to paraffin embedding. These samples were then studied alongside the original FTLD-TDP diagnostic blocks, which were fixed and processed and embedded 4 to 6 weeks after the donation had taken place. So 5 micron thick sections were cut from each block and were immunostained using the techniques that Yvonne and James has told you about, using antibodies for native TDP-43 and also for phospho-TDP-43, using a rabbit polyclonal antibody from Proteintech and a mouse monoclonal antibody from one of our longtime collaborators, Dr. Hasegawa in Japan. And in addition, for the FTLD-TDP cases, we immunostained the same regions with an anti-ubiquitin antibody to act as a standard against which to base TDP-43 immunoreactivity. So we also acquired bright field whole slide images using a Hamamatsu NanoZoomer S360 slide scanner at 20 times magnification. We then analyzed these images for relative stain intensity using QPath image analysis software, which is fairly common. And in short, what we did was region of interests were identified on the hippocampal sections and frontal sections, with care being taken to ensure that the regions corresponded between the different sections. A threshold was then applied to each region of interest to identify positively stained inclusions. Then DAB optical density measurements were obtained for all positive and negative cells within each region of interest. And then finally, the average negative optical density was subtracted from the average positive optical density to give a relative assessment. It's all relative, but a relative assessment of the staining intensity. So in the interest of time, I'm only going to show you the results from the hippocampus blocks. The immunoreactions in the frontal blocks were visible, but they were just slightly weaker. So this is better for visualization, really. And on the left here, you can see that the native TDP-43 antibody provided really high quality immunostaining of the inclusions in sections from the diagnostic blocks, which were those with a short fixation of the hippocampus. And then on the right-hand side, the native TDP43 antibody also provided really good quality immunostaining for TDP43 inclusions in sections from the blocks that were subjected to the extended fixation as well. So this antibody works both at short and extended fixation. The immunoreaction for ubiquitin showed staining patterns similar to TDP43, both in sections from the block subjected to short fixation and the sections from the tissue that was subjected to extended fixation. And this assured us that what we were looking at was the correct thing, as TDP43 inclusions in FTLD are ubiquitinated. Next, we looked at the monoclonal antibody, which was made in the lab of Dr. Hasegawa. And on the left, you can see that this antibody provided really high quality, very high quality immunostaining in sections cut from the diagnostic blocks of the hippocampus, i.e. those subjected to the short fixation. However, when examining stained sections from the blocks which were subjected to extended fixation, the immunoreaction appeared much weaker, which you can see on the right here. You can still see the inclusions, but they're much less strong as they are with the strong fixation. And we calculated that the mean DAB optical density for the monoclonal antibody was around 40 to 80% lower in extended fixation samples when compared with samples taken from the diagnostic blocks, those at short fixation. Then we looked at the commercially available polyclonal phospho-TDP43 antibody from Proteintech. And here the antibody provided really high quality immunostaining in sections cut from diagnostic blocks, which pinpointed the inclusions in the hippocampus. And then on the right-hand side, when examining stained sections from the extended fixation, the immunoreaction was still of very high quality. So it performed better than the monoclonal antibody. In this case, the mean DAB optical density of the polyclonal antibody was less affected than the monoclonal antibody and was only 25 to 30% weaker in the prolonged formalin fixation samples when compared with the ones at short fixation. And then finally, using the late cases, we ascertained that after a short fixation time, long-term storage of tissue as FFPE blocks had absolutely no detrimental effect at all on the immunoreactivity of phosphorylated TDP43. So the two antibodies used in the study performed very well and resulted in high-quality immunostaining. So it's clear that improving the diagnostic accuracy for TDP43 proteinopathies relies on really good quality immunostains. And likewise, research into these proteinopathies needs robust techniques to identify the phosphorylated inclusions associated with the disease. And in this study, we showed that phosphorylation at serine 409-410 can be detected after short and prolonged formalin fixation and also after long-term storage in paraffin. However, the sensitivity of the primary antibody is relevant to provide reliable immunoreactivity in the samples exposed to extended immersion in formalin. The polyclonal antibody from Proteintech performed better than the in-house antibody, the monoclonal antibody from Dr. Hasegawa. The higher overall sensitivity against the antigen due to the recognition of multiple epitopes by the polyclonal antibody possibly explains why it had a better performance. Regional variation in fixation may result in better preservation of inclusions in particular brain regions. So, for example, we found staining in the frontal gyrus to be slightly weaker than that in the hippocampus. We observed preserved immunoreactivity for phospho-TDP after long-term storage as FFPE blocks. However, there are some precautions to be considered to further improve immunoreactivity in blocks that have been subjected to long-term storage. So it's known that antigen degradation is more severe in unstained sections on slides when compared with FFPE blocks. So storage as blocks rather than as glass slides is probably the way to go to ensure good quality immunostaining. Similarly, some groups have shown that cold storage of FFPE blocks may preserve immunoreactivity better than FFPE blocks left at room temperature, though this method of storage could become very expensive if you've got a lot of blocks or you're part of a large archive facility like we are. So brain banks worldwide have been collecting tissue for decades, and in some cases, the original diagnoses will need updating upon the publication of new criteria and guidelines. For example, updating diagnoses using the late criteria has allowed us to maximize the use of old cases into research in this new entity. So we're now able, when people ask us for late cases, we're now able to give them late cases because we've gone back to the old tissue. When revisiting cases, we suggest that tissue preserved in formalin can be resampled when diagnostic blocks are exhausted, whilst accounting for this variability of staining intensity and regional variability. But we would say that extra sampling and processing to FFPE blocks at the diagnostic stage might be most prudent. And similarly, diagnostic FFPE blocks that have been stored for decades should provide adequate immunoreactivity to accurately diagnose or do research on TDP43 proteinopathies. And that's basically it from me. All that's left for me is to acknowledge the people that have been integral to the work of the Brain Bank, Manchester Brain Bank. Top right, you've got Professor David Mann, who created the Brain Bank in the 80s. He retired in 2018. The Brain Bank's now in the capable hands of Professor Federico Roncaroli, shown here on the top left. The two people at the bottom don't need any introduction. You already know these guys. And I'd also like to say a big thank you to our funders, Alzheimer's Research UK and Alzheimer's Society, who continue to support brain banking in the UK through the Brains for Dementia Research Initiative. And of course, I'd like to say thank you to Proteintech for allowing us to speak today and also kindly providing us with one of the antibodies we used in the study. Thanks very much.
Amazing. Thanks so much, Andy. And James and Yvonne, if you want to get your screen up. I'll just quickly run through 1 minute of the products that we have available for IHC, and then I'll get into the questions. So without further ado, I'll quickly share my slide. So at Proteintech, we have a lot of products available for IHC, including various prep and detect kits for running everything yourself, as well as obviously all the antibodies that we have for IHC. We also have hydrophobic pack pens that I know Yvonne mentioned as something that's really useful for preventing all that buffer falling off the slide. We have our directly conjugated antibodies for immunofluorescence. These are conjugated against our Coralite Plus fluorophores, which are very similar to the Alexa Fluor Plus, if you're not aware of these as a product. We also have our flexible antibody labeling kits. So these are a really useful reagent in terms of labeling your antibody with a polypeptide. It can do it in as little as 10 minutes. It's not covalently conjugated, so you don't have to do any buffer exchange. And we have over 7 fluorophores, as well as all these different specificities and HRP and biotin. So if you're interested, we don't have time to explain any more today, but do visit our website and you can see more information for that here. In addition to our flexible kits, we also have our new products. These are our recombinant secondary antibodies. We have these again on we have these again for our Coralite Plus fluorophores, as well as our polymer HRP. And because you attended our webinar today, you are able to try a anti-rabbit or an anti-mouse polymer HRP of a recombinant secondaries for free, and you'll be able to see the form here. So if you'd like to try that for free, please do scan the form. And now I will get on to the questions. So again, thank you so much to Andy, James and Yvonne. That was a really, really incredible talk. I've been messaging my colleague and we've been talking about how amazing the content is of your slides and how clear it was. So thank you so much. And I think just by the level of engagement and the number of questions that we've had, I think is indicative of how useful it has been. So there's quite a few questions, so I will go through. So one of them is from Peter Jessup, and he asks, have you ever used a different clearing agent other than xylene, such as chloroform? You are all muted.
You're muted, James.
How's that? Is that better?
Yeah.
Great. Yeah. No, we haven't.
No.
We have enough trouble getting rid of solvents as it is. And so, no, we've not. Okay. And someone asks, is there a solution if the brain tissue is very wrinkled while sectioning despite cooling the blocks?
It might be to do with process. I mean, there's 101 things it could be, but it needs to keep it cool, but maybe not too cool. So, like I said, warm it up a tiny bit. So it needs to be sort of in the middle. It is an art more than a science. I think hydration as well. So maybe just get a bit of water on the top. That's all I can suggest, really.
If I could just jump in for a second here, it's been a long time since I was putting sections in the laboratory, but what I used to do is I used to breathe on the block. So you actually warm the block up ever so slightly, and that removes some of the wrinkles as well. So where James is talking about cooling the block down and blowing on the section, sometimes slightly raising the temperature of the sections can also help flatten the slides out as well.
There is actually
Yeah.
another technique you can use. So if you get a glass slide and you put a bit of IMS on top, your wrinkly section, you place that on top, and that will fully stretch out, and then you take that section from there to water bath. That's something you could try. I've not done that in a long time, but that is something I know that people do.
Okay, that's really useful. Thanks, guys. And I know one question, there's actually quite a lot of questions about Sudan black bee and autofluorescence. And prior to this, we actually did a poll about what the people struggle the most with tissue IF and tissue IHC, and autofluorescence also came up as one of them. And I know when I've done a lot of IHC webinars, I've had people come to me absolutely desperate how to reduce autofluorescence in their clinical tissue. So one of the questions is, let's start with, is when do you use Sudan black bee? At what stage of the protocol do you use it?
So I've not thoroughly tested it, to be perfectly honest. But I've read quite a few papers because I'm going to be doing this in the future, going to be doing quite a bit of IF with the clearing and the 3D. And I think I'm not sure, I can send those papers to you to send out if you want to, but I think it was after antigen retrieval.
Okay, yeah, I think that's correct as well.
Yeah, Maria's just said she uses Sudan black before DAPI.
Yeah, I'm not sure how much of a difference it makes. I think I'd have to do an experiment really and try it in different places and see what difference it is.
I think there's definitely a lot of protocols out there as well.
Yeah, there's loads.
So if we're not quite correct, then please do confirm our answers. One of the questions is sort of more towards Andy and your prolonged fixation paper. And they're saying, what formalin concentration would you use for long-term storage or fixation? And would you try a lower concentration to allow for better epitope preservation?
Yeah, so we use 10% neutral buffered formalin. We just use that. So to be honest with you, I can't tell you whether using a lower concentration would be better, because we try and keep everything as standard as possible at the brain bank. So we just use 10% neutral buffered formalin. But it could be something that we look at in the future. You know, we're trying to expand some of these technical papers because they seem to really help people that maybe are at the beginning of their journey into IHC, and they're worried about things, especially from brain banks that have been kept in formalin for decades, and being able to show them that, yes, you can still see things after all that time in formalin, 10% neutral buffered formalin, then, I think it's a good thing for the community. But yeah, unfortunately, I haven't used any lower concentration of formalin, so I can't comment.
There's quite a lot of questions about antigen retrieval, and I know that's something that is incredibly useful for IHC. And one of them is, do you think that the baking temperature and the time would affect the binding of the antibody as well as the epitope sensitivity? And have you ever tried variations in baking time based on different brain regions?
We've certainly never tried it on different brain regions, but obviously over the years we have tried various methods of HIER. When we were doing the microwave, we were only microwaving for 10 minutes, not 20 minutes as we do with the steamer. None of it ever made any difference. Pressure cooker, microwave, steamer, they all came out really well. They all worked well. So I would say not, but it's not something we've investigated.
Okay, thank you. That's really helpful. And then, sorry, there's just so many questions, unfortunately. I'm just trying to find the, this is quite a good question about your study, Andy, and it's, how representative do you feel are your samples of the brain as a whole context of the disease? So I guess like a question about the epidemiology of the samples that you have.
You mean the samples, the regions that we took to use in the study? We basically chose the brain regions based on neuropathological criteria. So for late, for example, like I said in the talk, we use the regions of the brain that's used particularly in the published literature to neuropathologically diagnose that condition. So that's the middle frontal gyrus, hippocampus, and the amygdala. So we tended to pick regions that we knew we would go to reevaluate cases that would be in formalin for a long time.
Okay. And I think we have time for one last question. And I know this was something that I used to struggle with when I did a lot of IHC, and it was, we're really having trouble keeping 20 micron thick tissue from sliding off the slides during staining. Do you have any tips?
And my tip would be to get James to section it and put it on the slide.
Is there a reason why they're cutting it so thick?
I think often animal tissue use thicker slices. I think maybe it depends on the processing if they're not doing FFPE. So
Yeah. Make sure you're using coated slides that are in date. Make sure you bake them overnight. There's a thing sometimes when even if you bake them, sometimes water can be trapped under the section. Make sure there's no bubbles of water in there, because that can spread and that just sort of gets underneath the tissue and just lifts it off. So you can, like, if you get a tissue and fold it up to a point, you can sort of poke a hole in this with a sort of bag of water, and that sucks all the water out. Just get them as dry as possible. Maybe even use a desiccant if an oven isn't working, some silica gel, something like that.
That's a really helpful answer, actually. Thank you, James. I think we're at time there. There's still a few more questions, so we'll try and get around to them if we can afterwards. But if you have any pressing questions, and the contacts of all the speakers are up here. If you'd like to email Proteintech, it's events@pcglab.com. We will send everyone out a copy of the recording and also a copy of the slides, plus a link to the free sample. Apologies for the QR code being incorrect. That's on me. But I have now linked the correct link to the free sample if you're interested. But thank you so much to everyone who turned up. And again, a huge thank you to the Manchester Brain Bank team, Yvonne, James, and Andy for doing such an incredible presentation. I know I've learned so much, and I've been doing IHC workshops for 4 years now. So yeah, thank you so much to everyone. And I hope you enjoy your day, whatever time of day it is with you and wherever you are.
Amazing, thank you.